Hello. Thank you for joining us today to review a very important topic, biological risk assessment for IBC members. As members of an Institutional Biosafety Committee, you have an important role in ensuring that the research you review is conducted safely and responsibly. While the principal investigator is ultimately responsible for the safety of their team, biosafety may not be their area of expertise. That's where you and your fellow IBC members come in. You are the biosafety experts at the table. And to serve in that role effectively, there are a few key things you need to know. What are our objectives today? In this brief session, we'll cover the foundations of biological risk assessment as they relate to the IBC and the NIH Guidelines for research involving recombinant or synthetic nucleic acid molecules. We'll start by clarifying the difference between hazard and risk, and then take a closer look at what a risk assessment actually involves. Along the way, we'll address key questions such as: what do the NIH guidelines require in terms of IBC risk assessment? And what does the process look like in practice? From there, we'll walk through the core steps, identifying hazards, assessing risk, and applying risk mitigation strategies. Finally, we'll briefly explore how risk assessments may vary depending on the type of research reviewed by the IBC. And just to set expectations, this is not a full course. Risk assessment can be the subject of multi-day workshops. Today, our goal is a focused overview. When you finish this training, there are a few important points to remember. First, don't just look at the hazard of an agent, be sure to assess the overall risk of the research. Second, understand how to bring that risk to an acceptable level by applying biological containment principles. And third, know where to find the best resources to help you do this. You don't need to memorize every detail. What matters is knowing where to look for the information when you need it. Generally speaking, risk management is a series of actions, a process to identify and evaluate risk. A comprehensive risk assessment covers the first steps in the broader risk management process. In biosafety, this means identifying hazards, asking what could happen and how, evaluating risks, considering how likely is it and how severe could the consequences be, determining controls, deciding what measures can reduce the risk to an acceptable level, implementing and reviewing, putting controls in place, then checking if they work and adjusting as needed, measuring and controlling, reviewing effectiveness, monitoring outcomes, and adjusting as needed. By understanding where the greatest vulnerabilities lie, organizations can take targeted action, whether that means improving safety protocols, enhancing worker training, or even redesigning spaces to minimize contact with harmful agents. Risk assessment doesn't just protect individuals. It also helps laboratories operate more smoothly and efficiently by ensuring that resources are directed where they're needed most. A thorough risk assessment also demonstrates that an institution takes safety and accountability seriously, which is essential for meeting both legal and ethical responsibilities. And just as importantly, it builds confidence among staff, knowing that measures are in place to safeguard their health. So let's begin with hazards, risks, and risk assessments. Before we can talk about risk assessments, we need to distinguish between two terms that we'll be using often, and that are sometimes confused with one another: hazard and risk. A hazard is any substance or agent that poses a potential threat to health. Risk is the likelihood or probability that the hazard will actually cause harm under specific conditions. Risk depends both on the chance of exposure and on the severity of the harm it could cause. Hazard refers to any substance that has an intrinsic property with potential to cause harm. Take a shark, for example. A shark is inherently hazardous. If I ask you to think about the hazards of a shark, what comes to mind? You might say, it's a wild animal. It has rows of sharp teeth, a powerful jaw. It's an apex predator. But here's the important point. While you may think of pain or shark attacks, those are risks, not hazards. There is no actual risk of harm from those hazards, sharp teeth, or predatory instincts, unless you put yourself into a situation with the shark. Now, as soon as you jump into shark-infested waters, that's a different story. A shark attack only becomes meaningful once you leave the beach and enter the water. The risk of harm comes not just from being near a hungry, toothy shark, but from the probability that the shark will harm you, whether by biting, ramming, or even brushing against you and disrupting your air supply. And remember, just because you get in the water with a shark doesn't guarantee that you'll be harmed. It is the probability of harm, from any number of interactions with the hazard, that equal risk. Risk is not a fixed value. It reflects the interaction between how likely an event is to occur and how severe the outcome could be if it does. The more likely something harmful is to happen, and the more serious its effects, the higher the risk. That said, even a low-likelihood event with severe consequences may still require strong controls. And a high-likelihood event with mild consequences may still demand attention because of how often it occurs. Our goal is to identify where the risks lie and then apply appropriate measures to reduce both the chance of exposure and the potential impact. There are many types of biological risk. These include accidental infection, a toxic response, or an allergic reaction due to exposure to a biological hazard. Risks can also involve accidental release into the environment, leading to negative impacts outside the lab. Then there are biosecurity risks, such as unauthorized access, theft, or misuse by someone intending to cause harm. And finally, we sometimes have to consider the perceived risk of a hazard, which can influence how research is received by the public. For example, imagine a lab working with a genetically modified bacterium that produces a toxin. If a researcher accidentally sticks themselves with a contaminated sharp, the likelihood of exposure is high, and the consequence could be serious illness, resulting in a high overall risk. If that same strain is improperly disposed of and escapes into the sewage system, it becomes an environmental risk. And if it's stored without proper security measures, it could also present a biosecurity risk. A key point to remember is that risk management strategies are designed to minimize risk to a manageable level. But risk can never be completely eliminated. Going back to our shark at the beach, the only way to eliminate risk and avoid any possibility of harm from the shark is simply not to go in the water. The NIH Guidelines devote a considerable amount of attention to risk assessment and risk mitigation. In fact, this makes up the bulk of the document. It begins with Section 2, Safety Considerations, and continues through several appendices, including B, F, G, H, I, K, L, and M. When research activities fall under the NIH Guidelines, the responsibility for biological risk assessment rests squarely on the institution, and specifically on the IBC. Section 2, titled Safety Considerations, outlines the expectations for how institutions and IBCs ensure that this research is conducted safely and responsibly. This is the heart of your role as an IBC member. The committee is responsible for evaluating not only what the research is, but how it's done, who is doing it, and whether the containment measures are appropriate for the risks involved. Every research protocol you evaluate is, in some way, asking the same questions. Is this safe? Are the right controls in place? Is the risk managed? Truly accurate and reliable risk assessment cannot come from a single viewpoint. Effective risk assessment brings together input from multiple perspectives, including laboratory staff, biosafety officers, technicians, and facility managers. Each contributes unique knowledge, whether it's about daily lab practices, the behavior of specific biological agents, or the limitations of the physical workspace. The process typically begins with the principal investigator. As the person responsible for the project, the PI plays a critical role in identifying the biological agents involved and proposing initial safety measures based on the nature of the work. But those proposed measures should always be reviewed and discussed by the broader team to make sure all risks are thoroughly considered and appropriately managed. This collective approach helps identify gaps, challenge assumptions, and ensure that safety measures are practical, effective, and suited to real working conditions. In other words, while the IBC is charged with oversight of this research, it depends on the PI and their team to provide accurate, up-to-date information. As mentioned previously, the guidelines state that in addition to assessing the hazards or the biological agents, the IBC must review containment levels, facilities, procedures, practices, training, and expertise. This is why the continuing review and amendment process is so important. Even seemingly small modifications can introduce new risks that need to be evaluated. This is why a risk assessment should be performed at key moments during the lifetime of the research project to ensure ongoing safety and compliance, and a change in any of these variables has the potential to alter the biosafety profile of the work. Up to this point, we've talked about what the NIH guidelines say and how the IBC fits into that framework. But here's the key connection. Risk assessment is the tool the IBC uses to actually carry out that responsibility. When you're reviewing a protocol, it's not enough to stop at the hazard of the agent itself. The guidelines expect us to look at the whole picture: the procedures being performed, the people doing the work, and the environment where it's happening. That's why risk assessment is so central. It allows you, as an IBC member, to make an informed, confident decision about whether the research can be done safely and responsibly, and to ensure the institution stays in compliance with NIH expectations. So with that foundation in mind, let's shift from why risk assessment matters to how it's actually performed. The NIH guidelines provide a framework to identify and mitigate risks when working with recombinant or synthetic nucleic acids. They guide researchers in considering the host organism, vector, inserted genetic material, and potential hazards. Based on these factors, the guidelines recommend appropriate biosafety levels and containment practices to protect people, the environment, and research integrity. Our task is to perform a comprehensive risk assessment. To do this, we have to consider the context of the entire experiment. Each of these factors, including worker experience, adherence to safety practices, the type of procedures performed, and the design of the lab, affects both the likelihood and the severity of a harmful event. Let's start at the beginning. We are assessing biological materials. At what point do those materials become biohazards? A biohazard is a potentially infectious agent or hazardous biological material that presents a risk or a potential risk to the health of humans, animals, plants, and/or the environment. The list includes microorganisms, prion, biological toxins, genetically modified organisms, as well as plants and animals injected with biohazards. Once you have identified your biological materials and which agents may pose a hazard, it's time to see if there is a risk group classification for the agent. The pathogen's risk group classification system is a good starting point for identifying relative risk of a biological agent. Found in Appendix B of the NIH Guidelines, the NIH categorizes agents using agent information like routes of infection, pathogenicity, stability of the pathogen, community risk, if there are vaccines or treatments, mortality rate, etc. The key takeaway: this RG number is just our initial clue. It's the beginning of our safety investigation, not the end. It is imperative to remember that risk group is not the end of the risk assessment, but the very beginning, and they do not cover all agents for all things. Risk groups are only for agents that have the potential to cause harm to human health. Agents that do not cause harm to humans, but may be harmful to plants or other animals, are not assigned a risk group. Classification does not include host or procedural factors. To repeat, risk group is not equal to risk assessment. Risk group is also not equal to biosafety containment levels. And finally, genetically modified agents do not have an assigned risk group. For example, in lieu of a risk group for a lentiviral vector, one needs to look at the parent organism, HIV, and move forward from there. And at the heart of it, the IBC exists because of the NIH guidelines on recombinant and synthetic nucleic acid research. This is our core charge. So, when we move into recombinant materials, we need to consider added factors that go beyond risk groups alone. When evaluating recombinant materials or genetically modified organisms, additional factors must be included in the risk assessment. The transgene or gene target. What function does the inserted gene serve? Could its expression increase virulence, toxin production, or environmental persistence? Is it sourced from a pathogen or from a host with known adverse effects? The vector. The properties of the vector are critical. Can it mobilize or recombine with other genetic elements? What is its host range and stability? The delivery method. The way genetic material is introduced affects both the likelihood of off-target changes and the risk of unintended spread. The host organism. Risk depends on where the transgene is expressed. Expression in a non-pathogenic lab strain may pose minimal concern, whereas expression in human cells or animal models may increase risk, especially if it alters cell behavior or impacts immune response. Each of these components, transgene, vector, delivery method, and host, adds complexity. Their interaction must be assessed carefully to determine if additional containment, control measures, or oversight are warranted. For example, when assessing the use of viral vectors in laboratory or clinical research, several key features of the system must be considered to understand potential risks. Let's start by identifying the parental virus. Its inherent pathogenicity, host range, and known transmission routes can influence the biosafety level required. Next, we need to evaluate the modifications made to the vector. Has the virus been attenuated? What is the nature of attenuation? Is it self-inactivating? Has its tropism, or cell targeting, been altered to extend the host range? The viral particles assembly process also plays a role. The use of helper viruses can introduce risks of recombination or contamination if not well controlled. A critical factor is whether the vector is replication competent. Even rare events leading to the restoration of replication can significantly change the risk profile. Finally, consider the nature of the genetic insert itself. Does it encode a protein that may alter host cell behavior, increase toxicity, or affect immune response? Based on the information about the vector and insert, what can we expect in case of exposure? Will the delivery system and administration procedure used in the study influence the risk? When working with human materials and cell lines, risk assessment must account for both biological origin and modifications made. Commercially available cell lines may seem low risk, but they often undergo limited screening, meaning undetected pathogens, such as latent viruses or mycoplasma, can still be present. Some human cell lines may exhibit immunogenic or tumorigenic properties. Primary human materials, including blood, body fluids, tissues, or surgical samples, pose additional risks. These materials may carry unknown or emerging pathogens, including, but not limited to, bloodborne pathogens like HIV, HBV, or HCV. Finally, when these cells are genetically modified in the lab, the modifications can alter risk. For example, introducing oncogenes, viral genes, or immune-modulating elements may increase hazard potential. Animal materials and cell lines may be hazardous, as well due to presence of the zoonotic agent that can potentially infect human cells. As noted in the previous slides, the risk assessment performed by the IBC involves understanding the details of the host, the vector, and the gene product, as well as the scale of the work being done. But the most important factor is identifying which procedures carry the greatest risk to personnel. That means reviewing the safety controls in place, engineering controls, PPE and occupational health measures, and making sure they are appropriate for the level of risk. Think of this as detective work. We need to ask, what exactly are we doing to it? A small experiment in a test tube is very different from large-scale production. What new features are being added? A gene product could introduce toxins, alter transmissibility, or have unexpected effects when combined with others. What's the escape plan, and how do we stop it? With things like gene drives, we need to consider worst-case scenarios of accidental release. Do we have a kryptonite? Is there a vaccine or effective treatment if exposure happens? Taking this holistic view helps us determine if the actual risk is higher or lower than the risk group suggests. The risk group is just a starting point. The real answer comes from context, procedures, and controls. It's important to assess the training of the research staff and the experience of the principal investigator to ensure the work is conducted safely and competently. Going back to our risk assessment review, remember that when reviewing a study, it's essential to consider the factors that influence both the likelihood and the consequences of an exposure. A lot of common lab procedures can generate aerosols, often without being obvious. Pipetting, centrifuging, vortexing, opening pressurized containers, or even removing caps from tubes can all release particles into the air. These aerosols may carry infectious agents and pose risks. Exposure volume and concentration matter. Working with high concentrations can result in doses exceeding the infectious threshold, thereby increasing risk. Unconventional routes of pathogen transmission, such as splashes to mucous membranes, may lead to unexpected or more severe disease outcomes. Procedures involving the use of needles, scalpels, and other sharps are always considered more risky, but even safety-engineered sharps can cause injury if used incorrectly. Careful procedural review is critical. The experiment should be considered step by step, and when in doubt, ask the biosafety officer to observe the process and provide feedback. The susceptibility of the host, including immune status or pre-existing conditions as factors, could be critical in case of exposure. Equipment, facility layout, and material handling logistics also play a significant role in overall risk. For example, equipment such as centrifuges, biosafety cabinets, and incubators should be placed to minimize cross-traffic and avoid disrupting airflow. Moving biological materials through shared spaces, e.g., hallways, elevators, or between labs, increases the chance of spills or exposure. When multiple research groups use the same equipment, like centrifuges or water baths, there's a risk of cross-contamination if users don't follow consistent cleaning and decontamination procedures. Maintenance work may introduce personnel unfamiliar with biosafety risks or create opportunities for accidental exposure if the equipment is not properly decontaminated beforehand. It is critical to assess whether effective disinfectants are selected for the agents in use and whether protocols are followed consistently. Inadequate decontamination can leave residual hazards behind. Improper segregation, handling, or disposal of biological waste can result in exposure to lab personnel, custodial staff, or even the public. When performing an in-depth assessment, the following questions must be asked. What is the intended outcome of the work? Are there any potential unintended consequences, such as unexpected effects on the host or ecosystems? How likely is accidental exposure or release of the biohazardous material? If something does happen, what are the possible consequences? Given all of this, is the level of risk acceptable for the institution? Can this study be approved as is, or does it require additional controls, or maybe reconsideration? Once we've answered these key questions, we can be confident we've assessed the research as fully as possible. Now it's time to look at the special considerations that may add another layer of complexity. Now that we've covered the general steps of a biological risk assessment, let's put that knowledge into practice. The NIH Guidelines recognize that some types of research carry unique risks that require additional consideration, including animal studies, human gene transfer clinical trials, and plant research with recombinant organisms. These research projects are covered in the guidelines under distinct appendixes. As IBC members, it is possible you will be asked to review these more complex cases. Let's explore how to approach them. Imagine a study involving mice infected with a recombinant viral vector. What risks should we consider? Is the animal transgenic, the introduced organism, or both? What is the specific animal model? How is it expected to interact with the biological agent? Species susceptibility, immune status, and behavior can all influence the likelihood and severity of exposure. Personnel may be exposed to hazardous biological agents or chemicals through aerosols generated during handling or procedures. There's also risk from bites, scratches, and needle sticks, as well as from shedding of the agent by the animal. Zoonotic diseases, those that can be transmitted from animals to humans, must be evaluated carefully, especially if working with non-human primates, rodents, or livestock. We also account for allergy risks, particularly to animal dander, urine, or bedding materials, which can develop or worsen over time. A potential loss of containment, such as an escaped animal, may lead to unintended exposure or environmental release. This underscores the importance of secure housing and handling practices. Lastly, proper decontamination procedures and hazardous waste management are critical. Animal carcasses, soiled bedding, and sharps require careful handling to prevent exposure and ensure regulatory compliance. Together, these factors form a complex but manageable risk profile, if assessed thoroughly and controlled appropriately. There could be a lot of questions behind. We are going to inject the animals. Some questions you need to ask during a committee review for a project including animal research may include: what animal models and procedures are proposed? Will animals be handled outside of containment? How might animal facility staff be exposed? What are the plans for housing, waste disposal, and carcass management? A thorough risk assessment integrates the animal model, the biological agent, and the facility practices to ensure both occupational and environmental safety. When reviewing plant research, many of the questions we ask during our animal protocol review will be similar. Imagine a study involving corn engineered with a recombinant DNA construct for pest resistance. What risks should we consider? Is the plant itself transgenic, the introduced organism, or both? What is the specific plant species? How is it expected to interact with the introduced DNA or organism and with its surrounding environment? Factors such as reproductive biology, pollination mechanisms, and potential for crossbreeding can all influence the likelihood and severity of gene spread. Personnel may be exposed to risks during planting, harvesting, or handling of transgenic material, especially if the plants produce bioactive compounds, allergens, or toxins. There is also risk of exposure through pollen, seed dust, or soil contact. Environmental spread of transgenes, through pollen dispersal, seed escape, or vegetative propagation, must be carefully evaluated, particularly for open field trials. The potential for volunteer plants to emerge in future seasons adds to this concern. We also account for ecological risks such as effects on pollinators, soil organisms, or non-target species, which may be altered by the introduced traits. A potential loss of containment, such as accidental seed dispersal outside of controlled areas, may lead to unintended persistence or environmental release. This underscores the importance of secure greenhouse conditions, physical containment, and field trial management. Lastly, proper decontamination and disposal procedures are critical. Transgenic plant waste, soil, and growth materials require careful handling to prevent spread and to ensure regulatory compliance. When working with plant material, environmental protection is the primary concern. When making a biosafety containment assignment, the IBC members should consider not only the source and nature of the introduced DNA, but also the inherited potential of the host to spread a new genetic feature. Some critical questions to consider. Will plants be grown in a growth chamber, greenhouse, or open field? If open field experiments are proposed, local environmental conditions shall be carefully reviewed. How will pollen or seeds be contained and prevented from dispersal? What is the plan for decontamination and disposal of plant waste and soil? Do the introduced traits, such as toxin production resistance, raise environmental or agricultural concerns if containment fails? Have local environmental factors like pollinators, wind, soil been addressed in the risk management plan? Plant research may not pose the same direct infection risks as animals or gene transfer, but it demands careful attention to environmental containment and agricultural impacts. The risks associated with human gene transfer studies fall into two main categories: risks related to the investigational product itself, and risks related to how that product is handled at the study site. As we've discussed, each vector carries its own intrinsic risks. These may stem from the virus it was originally derived from, the genetic modifications introduced, the genes or other material carried within the vector, as well as its targeting mechanisms and biodistribution characteristics. Handling risks, on the other hand, arise primarily during preparation and administration of the investigational product. These concerns focus on the specific processes required for product preparation and on the method of delivery during administration. It's important to note that universal and standard precautions apply to all procedures within the clinical setting. However, the exact personal protective equipment required will depend both on the study agent and on the route of administration. Additional factors to be considered include the choice of disinfection agents, post-administration care, and the proper disposal of the drug substance. When reviewing clinical trials that use biohazardous materials, such as viral vectors, bacteria, or cell-based therapies, the IBC will conduct a thorough review of the gene therapy trial. This assessment should address questions such as: how will the investigational product be stored, handled, and transported? What biosafety measures are in place at the clinical site? What PPE is appropriate for preparation and delivery? How will waste and unused product be disinfected and disposed of? Equally important is the training of personnel involved in the clinical trial. Handling investigational drugs often requires specialized knowledge and skills that go beyond standard clinical education. A risk assessment here must consider both the properties of the investigational product and the procedures required to administer it. Controls include proper facilities, staff training, and clear waste management plans. In each unique research area, there are different sets of questions to consider. For animal studies, we should focus on occupational hazards, exposure, zoonosis, allergies, containment. With plant research, we focus on environmental spread and containment measures. And in clinical trials, we need to consider investigational product properties, handling, and what happens when the participant leaves the clinic. Across all cases, IBC members apply the same risk assessment framework, but adapt it to the unique context of the research. Those examples show how diverse the work under IBC review can be, but the principles of risk assessment remain the same. Let's step back and bring it all together with a few key takeaways. Risk assessment is a science-based, systematic process used to identify, evaluate, and manage potential hazards in research. It must be continuously updated as new information emerges. Effective risk assessment requires clear communication and collaboration among investigators, biosafety professionals, and oversight bodies to ensure informed, responsible decision-making.